Thank you very much. So quite happy to be here. This is my third time, BioVis. Well, the mammalian brain is, as we know, is the most complicated organ in the known universe, and trying to understand is a very, very challenging mission. And we have been doing many in vitro and in vivo experiments in the lab, and still has been 100 years since Cajal started to find the morphology, the structure of the different morphologies of the brain, and we cannot understand it. So another approach that has been sparked by Henry Marker and the group is to build a 3D reconstruction of the brain and try to make in silico experiments using the recent technologies, high-performance computing technologies, in order to be able to glean some insights about the structure and function of the brain that we cannot actually reveal in the wet lab. So in summary, this is the blue brain simulation workflow. It involves more than 150 scientists from different topics and disciplines: neuroscientists, biologists, mathematics, computer scientists, visualization experts, and many others actually. But our concern here will be basically to this part. So once we do an experiment, then we build a model, and then we do some simulations using this model. And then we do some analysis, and potentially we use visualization in order to understand how does the model work, how's the structure of the different neurons connect to each other, and so on and so forth. For further details, actually, you can refer to a paper we published in Cell in 2015. It involves 82 co-authors, more than 18 contributors from different labs. So just to give a quick introduction about the model, we do reconstruct what's called the neocortical column, or that's the most basic elementary unit of the somatosensory cortex. And the column is basically composed of 6 different layers. So this is the way we actually do structure it. And our mission is to build a model of this column as a prototype or as a principle. So we have reconstructed 55 different morphological types from the somatosensory cortex of a 2-week-old rat. And we have actually found all the different structures of the different morphologies that we can integrate into this model. And also we found 11 electrophysiological types, and we integrated both the morphological types and the different electrophysiological types. We call them M types and E types, and we started to build the column that way. So as you can see here, we probabilistically insert the different neurons in 3-dimensional space, and then we try to find the touches of the synapses where they do connect. And at the end, we build one column. And once we build that column, we do some simulations, and we map those simulations. Here, we see digital reconstruction of an entire slice that's composed of 7 different columns stacked together. And we try to see how does the simulation react visually on the reconstructed column. So that was one part, which is reconstructing digital model of the brain, simulating it, and applying simulation data to see the electrophysiology of that model. On the other hand, we wanted to do some optical studies. We wanted to see how does the column would look like as if we can see it under the microscope. This requires actually to build volumetric models, not anymore the surface mesh models we use to visualize the simulations. So in the last couple of years, we have published 2 papers in BioVis. The first one was to simulate the light sheet fluorescence microscopy using low scattering tissue, clarified brain tissue basically. And the other one was the last year we found a model to do that for highly scattering brain tissue. So that was okay. My supervisor was so happy, but then he told me, look, I'm quite interested about that, but still it's not really my motivation. I would like to see you taking this tissue block and do it for an entire slice. Doing that for the first time wasn't really that easy to imagine. How can I do that? Because we do have many reconstruction artifacts for the basic morphologies. So I don't know exactly how to stretch it. Like in the beginning, I was just extracting a small block, and I know my limitation. If something was, if there was an issue or something in some block, then I could have fixed it. But doing it for an entire slice that way was extremely challenging. And the basic challenge is, for a volume, we need to have a watertight mesh. And this thing is not, it will never be guaranteed for all the complex arborization of the neurons. So that's what this paper is about. So our main contribution is to find the novel approach for building piecewise watertight mesh models of the neurons that we can later on use for building the volumes. We have actually made a novel algorithm for creating those meshes. And we have also implemented a high-performance distributed pipeline for creating those neuronal meshes that we can actually create thousands of them in a few hours on a single machine. And also, we had to build a voxelization workflow that can generate such huge volume in affordable amount of time as well. So we designed and implemented an OpenMP-based implementation, and we were able potentially to reconstruct an entire complex neocortical circuit that's composed of almost 210,000 neurons. Okay, for the summary of the workflow, because it involves many other details. So we have basically a morphology. This morphology is full of artifacts. In order to be able to build a mesh, we have to fix those artifacts. So we made a list, compiled a list of artifacts, and we managed to solve all the artifacts that could affect the voxelization workflow. We haven't solved all the artifacts, but we solved the artifacts only that would affect the reconstruction of the volumetric models only. So we managed to fix the artifacts, for example, the disconnected branches from the soma, any overlapping between the connections of the first order branches and the soma as well, and any intersecting branches. And then we started mapping the actual radii value of each section in the morphology, and then we started to build meshes. So we reconstructed the somata, which is the most complicated thing, on a physically plausible basis using Hooke's law and mass spring systems in order to take the exact shape of the real somata we can see under the microscope, because what we can get is just a 2D projection from the 3D reconstruction, which is, then we have a missing dimensionality, so we have to find a way. And at the end, we had to connect the branches in a tubular structure. So we then built the piecewise watertight mesh models, and then we started to do surface voxelization in order to create a surface shell of each individual neuron, but the entire process is done on the entire circuit, or the last process does on the entire circuit, so it doesn't consume that much time. And then we fill the extracellular space, and potentially we have we fill the intracellular space, but just an inversion operation. And that's how we reconstruct a volume at the end. So I know that some details might not be that clear here, so we can discuss them afterwards, actually. Okay, for the results. Okay, this is the reconstruction of the somata on a physically plausible basis. So basically, we pull a sphere until we get the actual shape of the somata. And we have selected 55 different exemplars of all the neurons that are reconstructed from the neocortical column, and we reconstructed the somata of all of them. So as you can see here, they do have totally different shapes, and they are way better approximation than models than the sphere approximation that people used in the past. And then for the meshes, we built them by connecting the different components, axons, apical dendrites, basal dendrites, and the somata into one single object. It's one single object that integrates all the different components of the mesh of a neuron, but they are not connected. They are integrated in one single object, and this will fulfill potentially our voxelization purposes. So comes here to a closer look, three different neurons, just random examples from the 55 from morphological samples. So we can see here the morphology, and then fixing the morphology and mapping it, and then reconstructing the different branches together, and then we connect them, and we integrate the soma into them. So they are all integrated into one single object, but they are not actually connected. So this will be seen here in the volume reconstruction. We have in A just a pure mesh model, and in B we do the surface voxelization. In C, we do the solid voxelization, and potentially we add the spines to it. So closer look, we will see that in B, since the components are not connected, we can see that they are still overlapping. But since we can use the solid voxelization workflow in order to do this inversion operation, which would potentially be the result in C, we don't have any intersections anymore. So the water tightness issue that was the main limitation for building such huge volume on a per neuron basis is not there anymore. We don't need it. But the potential result will be, as you can see here. So from that, actually, we started to, from one cell, we extended to a few pyramidal cells in B, and then more and more cells until potentially we had reconstructed an entire column. And finally, I fulfilled the desire of my supervisor, and I have shown him the result of reconstructing an entire slice for the first time. And he was actually quite happy after we were only able to build that small tissue block to extend it to an entire slice. So that result was quite, he was very happy about it, and I was actually happy as well. So trying then to exploit or to use this workflow to do some in silico experiments, we have applied the reconstructed model for simulating 3 different kinds of microscope. This is simulation of the bright field-like microscope with Golgi skinning. I don't know how many of you actually would be aware of that. But as you can see here, this image is a resemble of the bright field microscopy image we can see in vitro with the Golgi stain, and the image has been generated on a physically plausible basis using a physically accurate optical model. And then we use the model to reconstruct a fluorescent annotated tissue block, and on the left you can see that's an in vitro image, and on the right you can see the model we have derived back in 2015 that was presented in BioVis. We have applied that model to build an in silico physically plausible fluorescent image of the cortical tissue. And at the end, we have built an in silico Brembo, which many fluorescent proteins can express different kinds of cells, so they can randomly label different neurons in the tissue. And we have managed to reconstruct an in silico one. So those experiments actually can be extremely helpful to give us a way to find the responses of the different fluorescent dyes in silico, to address different characteristics of the cells, to use them for doing in silico voltage-sensitive dye imaging, many other fluorescence experiments that we can use in the tissue. And with that, I think I'm done. Thank you very much, and I'm open to any questions.
Yes, thank you. Well, there's ample time for questions, so please go ahead.
Hi, Marwan. That was really cool.
Thank you.
Have you heard of the Art and Science competition that's held here? Because as I was watching your animations, and then when I saw your brain bow, I mean, I could see those winning the competition. Have you thought of submitting them? I'm serious.
Yes, actually, we were about to do that. It was like 3 months ago. I discussed with my supervisor, but the issue is I got I had to finish my thesis, submit a few papers, and finish 2 workflows, and finish 2 more collaborations with other PhD students. So I had on my to-do list something like this. But we have actually considered the art and neuroscience. I can show you some images we have prepared for that. But my decision will be next year, actually, to submit.
Well, I have good news for you. I'm one of the co-chairs of that meeting, and the competition, as of today, is now open. So you have a whole year, or you have until June 1st.
Yeah, but I have submitted my thesis in 10 days. So once I'm done with that part, I will
You have until June 1st. You have a year, basically. But if I could also just say, we're also looking for committee members and for other submissions. So please look at Art in Science. I think a lot of people in here could really enhance that competition. Thank you.
I have 2 questions. First of all, amazing presentation. My question is, are you doing any sort of functional studies with your simulations? Are you simulating the action potentials?
Yes, so there are 2 different kinds of experimentation. The first one, as you can see in the spiking circuit, that's electrophysiological simulation based on surface meshes, but this is not the main concern of this work. We use the fluorescence model and the volumetric models of the tissue to build, to make in silico voltage-sensitive dye experiments. So we have done some experimentation, but we haven't yet published anything about that. And in the last 2 years, we we use the same model to do the in silico light sheet microscope, and we can actually do any kind of like different kinds of not any different kinds of simulating the activity of the neurons that way. So you have just check the concentrations and the other factors, and then you can do it. So that's some future work as well.
My second question is, so you were simulating healthy brains. Have you looked at simulating autism or schizophrenia where the column architecture is known to be disrupted?
No, the model is just limited now to the neocortical circuitry. We are extending it to work with the hippocampus, but I don't think, I don't know, maybe it could be a decision of my supervisor to do something different, but our main focus now is the somatosensory cortex and the hippocampus in the mammalian brain, to basically the rat brain.
And only in wild type brains, right?
Yeah. Yes.
Okay. Thank you.
Hello. I can see the use of the visualization in hypothesis generation, for instance, but I was wondering if, since most visualization methods would go in the direction of abstraction, the abstraction of what's actually happening, is there a way that the images that is generated through this method is more informative than a potential abstraction?
Of course, because these images, they do simulate the light tissue interaction on a physically plausible basis. In typical volume rendering application, you have just the emission, the basic emission optical model, which doesn't consider the absorption, which doesn't consider the reflection, which doesn't consider scattering. And these models in particular, they are much more complicated because they do inelastic scattering. So if you have different components, and you light the tissue with certain wavelength, you get a completely different response if you light it with if you enlight it with another wavelength. So the model, the optical model that's used here, it's a physically plausible model. So it takes actually to generate those images quite a few hours, one per frame.
Hi. Yeah, sorry. So you have 55 3D models of cells, correct? Or
55 exemplars, morphologies, yes.
Exactly.
Yeah.
And then you stack them.
No, these are just exemplars. What we do, we build from the basic reconstruction in the lab other models based on some statistical parameters. Based on these basic 55 skeletons, we build other structures, and then we integrate them into one structure, the column. So they are not exactly similar. They are just have some basic structure that form the main 55 reconstructed morphology, but they are totally different.
Okay, so the way that you basically connect two cells in terms of synapses, where do you get that data from?
We get the data of each individual morphology from the lab.
Okay.
We build a mesh model,
Okay.
and then we put them in some framework, and then we have special indexing framework that can get the touch detection or the synapse, where they can form the synapse, and then we do some 3 or 4 phases in order to find the right ones from the wrong ones, and potentially we have the model, the neocortical column model.
Okay. Okay.
So I would say it's just a complicated workflow. You can refer to the Cell paper, 2015 paper, and you find every little detail you can imagine about the reconstruction of the.
Okay, thanks.
Okay, thank you. So thank you, Marwan, for a nice talk and for the nice set of questions from the audience. So also welcome to the new people who just joined in. I